My name is Jelena Vulic. I am Associate Professor at the Faculty of Technology, University of Novi Sad in Serbia. I would like to share my presentation. Title of presentation is Fruit and Vegetable Waste Valorization. Uh, I would like to say something about the Faculty of Technology. Faculty of Technology, the University of Novi Sad in Serbia, was founded to give academic education in the sphere of food, chemical, and pharmaceutical engineering, as well as biotechnology. High level of academic education, scientific papers, projects, innovations, and patents have ranked the Faculty of Technology very high among other institutions. Faculty of Technology attaches great importance to international cooperation, which has been successfully achieved over the years through the numerous scientific projects, national and international projects, and it made network with researchers at other scientific institutions abroad, expanded areas of interest and research, but also provided financial support from various international funds. Like I said, my name is Jelena Vulic. I'm associate professor. I received my PhD on functional and antioxidant activities of beetroot pomace at the same institution. I'm professor at the Department of Applied and Engineering Chemistry, and my research areas belong to food chemistry, organic chemistry, antioxidants and additives, and aroma quality control in food chemistry. I'm author and co-author of over than 150 scientific papers, 4 book chapters. My articles have been cited more than 2,500 times, and Hirsch index is 31. I've been coordinating or participating in more than 20 national and international research and networking projects, bilateral projects, and etc. Fruit and vegetables are consumed every day, raw, minimally processed, or processed due to their nutritional and health promoting compounds. As we know, the world population is rapidly growing and changing diet habits. Also, the production and processing of fruits and vegetables is growing. Significant losses and waste are becoming a serious nutritional, economical, and environmental problem. The United Nations Food and Agriculture Organization has estimated that at least 1/3 of the food produced in the world is lost and wasted every year, and losses and waste in fruit and vegetables are the highest among all types of food. It is up to 60%. The fruit and vegetables waste is composed mainly of seeds, skin, rind, pomace, and those parts are still containing high amounts of potentially valuable bioactive compounds like natural pigments, polyphenols, dietary fibers, vitamins, enzymes, and oils. These phytochemicals can be utilized in different industries, including the food industry, for the development of functional or enriched foods, medicines, pharmaceuticals, and in textile industry. The use of waste is an important step towards sustainable development. Food losses are commonly the result of technical limitations in handling, such as storage, packaging, marketing, while food waste is usually the result of food discard. Losses and waste occur during all phases of the supply and handling chain, like harvesting, transport to packaging or markets, classification, storage, marketing, processing, and at home before or after preparation. Fruit and vegetable losses and waste do not represent only wasting of food, but also wasting of land, water, fertilizers, chemicals, and energy. This is a big environmental problem as they decompose in landfills and emit harmful greenhouse gases. Followed by household garbage, fruit and vegetable processing units commonly produce the highest waste into the environment. During fruit processing, solid like peel, seeds, stones, and liquid waste like juices and wash water are produced, while their storage in non-appropriate conditions could bring flies and different insects or animals in processing areas. Also, fruit and vegetable waste are subject to microbial degradation and autooxidation due to high water content and active enzymes, and consequently uncontrolled oxygen consumption and greenhouse gas emissions could cause environmental problems. Circular economy is a term, just a second, is a term for industrial economy that is producing no waste and pollution by design or intention, and in which material flows are of two types. First type is biological nutrients designed to re-enter the biosphere safely, and technical nutrients designed to circulate at high quality in the production system without entering the biosphere, as well as being restorative and regenerative by design. Circular economy in the food industry is based on the valorization of waste, changing markets, technologies, and institutions through innovation. Valorization pathways are diverse, depending on the source, resource, and the end user, with the general idea of making a profit by exploiting waste. Byproducts of industrial processing of fruit and vegetables, like, for example, grape pomace, citrus pulp, apple pomace, carrot peel and pulp, tomato waste, and beet waste, are a rich source of dietary fiber. Polyphenolic compounds and natural pigments, and after processing, are treated as waste. Besides nutrients and bioactive ingredients in functional food, byproducts from food processing side streams managed adequately can be used like as bioabsorbents, additives, animal food, microorganism, growth substrate, fertilizer materials after composting, or as energy sources, substrates for biofuel production, and etc. Moreover, prebiotics from this waste material, in combination with antioxidants, are often associated with the possible therapeutic applications in formulas that enhance the human health by improving the intestinal environment and metabolic processes with insignificant side effects. Usually, only the fruit and vegetable flesh or pulp is consumed, producing significant amounts of waste, but many studies prove that significant amounts of health beneficial compounds are still, like we said previously, in seeds, peels, and other components of fruit and vegetables, which is often not used. Waste from fruit and vegetable is known as a rich source of, like we previously said, valuable bioactive compounds, natural pigments, phenolics, dietary fibers, sugar derivatives, organic acids, minerals, and others. This waste could be used to extract and isolate bioactive compounds that can be used in the food through food enriched with health-enhancing substances, pharmaceutical, cosmetics, and textile industries. Also, these bioactive compounds possess beneficial health attributes like antibacterial, anti-tumor, antiviral, antimutagenic, and cardioprotective activities. Applications of fruit and vegetables byproducts could be divided into two categories: those with technical purposes, which include the improvement of shelf life, safety, stability, sensory quality, and etc., and those with biological purposes, which aim to enhance health promoting effects. Now, I would like to say a little bit more about phenolic compounds. It is known that phenolic compounds are among the largest classes of bioactive compounds with diverse and important biological functions. Phenolic compounds are secondary metabolites synthesized in plants and possess one or more phenolic rings with one or more attached hydroxyl groups. Depending on the strength of phenolic rings, they are classified into several groups: into phenolic acids, flavonoids, stilbenes, and lignans. They are found naturally in fruits, vegetables, nuts, coffee, tea, seeds, and in their agro-industrial byproducts. These compounds offer numerous health benefits, well-known antioxidant activity, then anti-cancer, anti-mutagenic, antimicrobial, and other bioactive properties. A growing number of research evidences prove that the content of phytochemical compounds is higher in peel and seeds with respect to the edible tissue of fruits and vegetables. Many factors affect the concentration of phenolic compounds, like these factors usually include environmental factors like soil, sunlight, rainfall, various farming methods, plant varieties, tree fruiting, as well as biochemical factors such as degree of ripening, storage, and cooking methods. The level of antioxidant activity of different phenolic compounds varies according to their chemical structure. The antioxidant property of phenolic compounds stems from their hydroxyl groups and conjugated aromatic systems, and it depends on the ability of phenolic compounds to give electrons to trap free radicals by forming stable phenoxyl compounds. Antioxidants can be categorized in many different ways. Well known is in natural and synthetic, then polar, non-polar, enzymatic and non-enzymatic, endogenous and exogenous, and also by the mechanism in which they are involved. Antioxidants primarily exhibit activities based on three mechanisms: hydrogen atom transfer, singular electron transfer, and metal halation. Endogenous antioxidants are primarily enzymes, such as, for example, superoxidase mutase, catalase, glutathione reductase, and glutathione peroxidase. While non-enzymatic endogenous antioxidants, such as glutathione and lipoic acid, are produced and products of the body's metabolism. The first-line defense antioxidants, enzymatic, convert reactive superoxide and hydrogen peroxide into water and oxygen. The non-enzymatic antioxidants can act as a second-line defense against reactive oxygen species by rapidly inactivating radicals and oxidants. The enzymatic antioxidants further act as the third-line defense involved in detoxifying and removal. Dietary antioxidants such as vitamins, carotenoids, polyphenols, flavonoids, and bioflavonoids are exogenous antioxidants that have in vivo activity. Oxidative stress. Oxidative stress is defined as the imbalance between the occurrence of reactive oxygen, nitrogen species, and cellular antioxidant defenses. Oxidative stress is a result of excess of reactive oxygen or nitrogen species which occurs due to a lack of counteraction by cellular antioxidant systems. Increased oxidative stress can have severe consequences in biological systems, including molecular damage, such as nucleic acids, lipids, and proteins, which can potentially severely impact health. Damage to biomolecules or the induction of several secondary reactive species due to oxidative stress ultimately leads to cell death, apoptosis, or necrosis. It was proved that oxidative stress is connected with more than 100 diseases, including cardiovascular diseases, cancer, hypertension, diabetes, neurodegenerative diseases, aging, and etc. Contrary to their harmful effects on health, reactive oxygen or nitrogen species can have beneficial effects depending on their function, location, and amount. For instance, superoxide and nitric oxide radicals at low or medium concentrations are involved in cellular responses and participate in signaling pathways. Hydrogen peroxide, formed by various oxidase enzymes and the action of superoxidismutase, allows its use as an important signaling molecule. Also, it is substrate for regenerating further reactive species such as hypochlorous acid. Reactive oxygen species are also involved in immunological responses, degrading xeno compounds and organisms through phagocytosis. Reactive oxygen species are oxygen-containing molecules, including radicals, like the superoxide anion, and non-radicals, that greatly vary in their chemical abilities, such as diffusion in living cells and chemical reactivity with biomolecules. Reactive oxygen species examples include singlet oxygen, superoxide, hydrogen peroxide, and hydroxyl radicals. Singlet oxygen is the highest energy spin state of molecular oxygen. In contrast to molecular oxygen in ground state, the two valence electrons are paired in an antibody orbital. Singlet oxygen is therefore only generated when molecular oxygen is energized via radiation. In contrast to other reactive oxygen subspecies, no electron transfer does occur during this process. Singlet oxygen is very reactive towards organic compounds and plays a damaging role in biological systems, for instance, by involving in the oxidation of LDL cholesterol, which can lead to cardiovascular diseases. Moreover, increased reactive oxygen species can trigger mitochondrial DNA mutations as well as promote uncontrolled proliferation and carcinogenesis. The balance of harmful and beneficial effects of free radicals is crucial for life processes, and antioxidants play an essential role in achieving this balance. And there is a small photo of a balance of free radicals and antioxidants. It's usually shown like that, like the balance. Antioxidants assays. I would like to say something more about antioxidant assays. There are different mechanistic pathways. One of them is electron transfer-based assays. In these assays, a single electron transfer occurs between the antioxidant and substrate, which is measured to assess the potential of the plant's secondary metabolites. Examples: assays like cupricune reduction antioxidant capacity, then DMPD test, then ferric reducing antioxidant power, known as PrEP, ABTS radical cation decolorization assays, and etc. The second mechanistic pathways are hydrogen atom transfer-based assays. In these assays, hydrogen atom transfers from the antioxidant to the substrate, and assays such as oxygen radical absorption capacity or trap methods like total radical trapping antioxidant Parker methods are examples. Then there is a mixed, like electron-hydrogen atom transfer-based assays. In these assays, the hydrogen atom transfer occurs via two-step mechanisms. Also well-known antioxidant assays mechanistic pathways is metal halation-based assays. In these assays, antioxidants halate with transition metals. Ferrous ion and cuprous ion halating activity are examples. And there is lipid oxidation and reactive oxygen and reactive nitrogen species scavenging activity assays. These assays are based on the ability of antioxidants in reducing, preventing lipid oxidation and scavenging of radical species. Well known is beta-carotene assay, then FTC, thiobarbituric acid method, and others. Here you have the presentation some more examples. Then there is enzymatic antioxidant assays, antioxidant enzyme systems that catalyze reaction to counterbalance free radicals and reactive oxygen species, include superoxidase mutase and catalase. And also there are some examples of these enzymatic antioxidant assays. I'm mentioning them because it is known that fruit and vegetables are rich sources of antioxidant compounds, and of course, antioxidant assays have to be mentioned. And I wanted to tell you something more about them. Now, to continue with the natural colorants, like we said that these fruit and vegetable wastes are rich sources of phenolic compounds and also rich sources of natural colorants. Natural pigments present in fruit and vegetable byproducts can be used as a natural colorants in industries that we previously mentioned, like carotenoids, betalains, anthocyanins still remain in fruit and vegetable byproducts in large amounts. It is proved. Many scientific studies have proved this. In recent years, there has been an increased interest in the development of non-toxic and environmentally friendly natural dyes, since there is awareness of healthy lifestyle is growing day by day. That is why consumers are turning their attention to natural and safe food, and frequent discoveries of harmful effects of synthetic pigments on human health have led to public interest in natural pigments as an alternative to synthetic ones, and resulting in increased research in the development of natural colorants. Now something about betalains. Beetroot, Beta vulgaris, contains a number of bioactive compounds, phenolic compounds like flavonoids and phenolic acids, but also colored ones, natural pigments, betalains, red-violet beta-cyanins, and yellow-orange beta-xanthines. And those compounds can exhibit health-promoting effects, mostly due to their antioxidant properties. But betalains are commonly used food colorant and additive in various food products like juice, which is candies, yogurt, and betaline pigments derived from beetroot are a natural alternative to synthetic red dyes. In this context, betalines from beetroot are the most promising, not only as colorant, but also because of their free radical scavenging and reducing properties. Other reported health benefits are antimicrobial, anti-cancer, anti-lipidemic, anti-inflammatory activities, as well as hepato and neuroprotective effects. Many scientific studies, including studies in our laboratory, proved very high amounts of betalines remaining in beetroot byproducts. You can see in this photo how beautiful is this natural beetroot color. Carotenoids. Now, something about carotenoids. Carotenoids stand as the major group of compounds used as coloring agents, which can provide better sensorial, nutritional, and antioxidant quality of the final product. In general, carotenoids in food are classified into carotenes and xanthophylls. They are widely distributed fat-soluble pigments which naturally exhibit red, orange, or yellow color of fruit and vegetables. The biological functions of carotenoids are influenced by their chemical structure. The basic molecular structure of most carotenoids consists of a polyisoprenoid carbon chain with a series of conjugated double bonds located in the central portion of the molecule. This feature permits effective delocalization of electrons along the entire length of the polyen chain and provides carotenoids their pigmentation, light harvesting potential during photosynthesis and chemical reactivity. Another structural feature of most carotenoid molecules is the presence of cyclic end groups. Recent interest in carotenoids mostly arises from their health benefits. Some carotenoids serve as a precursor of vitamin A, and it is known that vitamin A is essential for vision, reproduction, and bone health. In addition, vitamin A plays an important role in the immune system function. However, the use of betalains and phenolic compounds in food industries and food products has been restricted because these bioactive compounds are very unstable under different conditions, such as pH, light, temperature, and the presence of oxygen. Changes in the chemical structure of bioactive compounds could happen and lead to the loss of their activity and potential health benefits. Also, carotenoids are very susceptible to degradation. In fact, the high number of conjugated bonds, double bonds, makes carotenoids prone to auto-oxidation and isomerization during food processing and storage. Also here, you can see a very nice natural color of carotenoids. Encapsulation technique has been intensively studied as a promising method for improving stability to preserve the biological activity and to ensure controlled release of the active compounds. In the food sector, encapsulation is a useful tool to improve delivery of bioactive molecules like antioxidants, minerals, vitamins, carotenoids, and living cells like probiotics into foods, keeping the bioactives fully functional. In the process of encapsulation, one material or a mixture of materials, which is known as core, is coated with or trapped within another material, a system called the wall material. Many encapsulation technologies are available, like spray drying, spray cooling, spray chilling, conservation, extrusion, fluidized bed coating, molecular inclusion, and freeze drying. Also, a range of edible materials can be used as wall materials for encapsulates, polysaccharides, proteins, fats, and waxes. Accordingly, by encapsulation, bioactive compounds as core materials can be stabilized using different matrices. The selection of appropriate carrier material, encapsulation technique, and encapsulation conditions are critical issues, which require consideration for the process of encapsulation. Encapsulation efficiency, a term which is also known as loading efficiency, is a numerical measure of the amount of incorporated core material in microparticles, which is generally expressed as the percentage of core material encapsulated relative to the amount of initially added core material. Encapsulation efficiency is one of the critical properties of microencapsulation that have important influence on subsequent application. Microparticles with high encapsulation efficiencies are often desired as less carrier materials are required and less net amount of encapsulated products could deliver the required amount of core material. This is the list of some representative projects. Some of them I will explain a little bit more, like the first one where we use the byproducts from fruit and vegetables, and also in this one we use the waste from the corn, sweet corn waste, in the production of after producing baby's food. And here's the list of some others representative projects where I was involved. The first one, which I would like to say more, is the title is nutritionally and functionally enhanced dairy products using fruit and vegetable byproducts. In this project, beetroot pomace extract, so waste after waste material after beetroot processing, and pumpkin pomace, also waste material, were mixed under the optimal experimental conditions in terms of the best encapsulation efficiency to scale up the process and translate it to the industrial production. Obtained encapsulate at pilot scale showed very good antioxidant, anti-hyperglycemic, anti-inflammatory properties, activities, as well as bioavailability after in vitro digestion tests. Also, tested antimicrobial and physicochemical properties showed beneficial potentials for application in food industry. After that, dairy functional products were processed first in laboratory and then after that in industrial level, and bioactivity and product quality like microbial, sensory and rheological testing of yogurt enriched with optimal beetroot encapsulates made, like we said, from beet pomace and pumpkin pomace were tested, and also stability, safety, and bioactivity of functional dairy products during their standard shelf life were investigated. Produced yogurt with bioactive compounds at industrial settings proved beneficial potential for application in food industry. The product prepared from commercialization is presented at the following web page. This is the web page, and we called this healthy product Beat It, which is strong and healthy. Why? Because just 1 cup of Beat It offers 100 more proteins than the plain yogurt. Then it contains all 9 essential amino acids. Then antioxidant value of 200% RDA vitamin E. Then proteins and antioxidants are all plant-based, and we added 0 sugar, and it still this product contained original sour tastes. Also, this product is rich in antioxidants, antioxidant compounds like phenolics and betalains. Here's something about our story, and also the photo of participants in this project, innovators, and their names. So the products that we made first in laboratory and then in industrial level, we called it Beat It. It is functional yogurt enriched with encapsulate that contains beetroot waste and also pumpkin pomace waste. The second one that I would like to tell you more, the project title is New Concept of also yogurt, but formulation, but now with pumpkin byproduct extract. The aim of the project was valorization of pumpkin byproduct through their application and creating a new value-added product, but now yogurt with pumpkin byproduct extract. Using solvents with different polarities proved the most efficient extraction solvent for targeted compounds. Those target compounds were polyphenols and carotenoids. And in order to protect targeted compounds from degradation factors, we used two methods for encapsulation. One was freeze drying. No, here we used only freeze drying technique, and in the other project we used more techniques. Encapsulation was proposed as a successful method to improve the bioaccessibility and bioavailability of the bioactive compounds. High quality, competitive, and economically acceptable functional yogurt was prepared. Here is the photo of this yogurt. Stability of targeted compounds, color parameters, and microbiological safety were evaluated as critical parameters to provide a high-quality yogurt during the storage period. We made a microbiologically, sensory, and medically valuable functional product which could easily reach the market and meet consumer demands. First, we made extraction optimization, then encapsulation optimization, then we tested encapsulate, and we tested the viability of making functional yogurt with optimal encapsulate was added to yogurt after fermentation at 5 different concentrations. In order to make the final decision of the level of yogurt supplementation, the following criteria that were used were sensorial characteristics. It is very important, of course, for the consumer, like appearance, smell, flavor, aroma, texture, and aftertaste. Then the level of supplementation with bioactive compounds. Then cost effectiveness of yogurt supplementation, which is also very important for the users. And at the end, final product, functional yogurt testing. So here we used pumpkin products in this project. One more project that I would like to tell you more about is the project title was chemometric modeling of functional characteristic of corn-enriched zeaxanthin foods. Here we used sweet corn byproduct extract. And now in this project, carotenoid-rich encapsulates based on sweet corn byproduct extracts that was rich in carotenoid pigments, especially rich in zeaxanthin, was obtained by the freeze drying and spray drying techniques. So here we used two encapsulation methods, and we also used 4 different raw materials: soy and pea proteins. We used maltodextrin and inulin. For the assessment of the stability of the obtained encapsulates, the physicochemical characteristics of all 8 types were determined. The other aim of this study was to use classification and ranking tools, so statistics, in order to detect grouping amount the investigated encapsulates and to detect the most suitable encapsulates for the assessment of conceivable applications as food additives. The most suitable raw material and encapsulation technique for the assessment of sustainability in food products was produced by freeze drying, so freeze drying method, and pea protein as a raw material. In laboratory conditions, we also made tagliatellas, so pasta, enriched with corn encapsulates, and we prepared it. The preparation of tagliatelle involved the substitution of part of the flour, 10%, with a functional additive. The content of total carotenoids was determined before and after cooking the tagliatelle. The highest carotenoid retention was shown by tagliatelle with freeze-dried encapsulated and pea protein, like we previously said, and substituting part of the flour with encapsulated zeaxanthin from corn, sweet corn, in the production of tagliatelles creates the possibility of improving nutritional functional and sensory properties, as well as the possibility for further improving the quality of the raw material and the production process, so to create new healthy products. And here's the photo of these 4 encapsulates that we prepared in this project. And like I said, we used sweet corn byproduct with very nice yellow color. This I just made small list of representative publications where different fruit and vegetable waste materials were used. Here, for example, we used peach pomace. Here we used beetroot byproduct. Then here we used sweet corn byproduct. Here we used red pepper waste in this paper and in this research. Here we used the horned melon peel, then pumpkin waste, then different parts, including, for example, peel of pomegranate, wild and cultivated. Then here we used also pumpkin waste. In this paper we used horned melon, different parts of horned melon, and also here carrot waste was used also to enrich pasta. And there are, of course, a list of many more, which I did not put here. You can look on the internet. Now, I would like to thank you for your attention. I hope the presentation was interesting and easy to follow. If you would like to contact me, this is my email. I wrote it here, and like I on the beginning I said my name, and that was it. Thank you once more.